Hey everyone, my name is Nick Delisi, if you haven't met me before, and I am the lab manager at St. Tammany Parish Mosquito Abatement, just about 30 miles north of New Orleans in Louisiana. And I'm here to talk today about resistance, its mechanisms, and how not mostly to start a statewide surveillance program for resistance. Let's begin with the basics of what insecticide resistance is. I'm sure everyone is very familiar with it at this point, but just in case you aren't, the way that I describe it is a loss of insecticide efficacy due to selection of heritable traits, which is a lot of gibberish, so let's go through an example. So right now in your population, you have resistant mosquitoes. You might not think that you do, but they are there just because of random mutation and chance. So I'm going to characterize those mosquitoes. They are going to be our green ones. You can see they make up the minority compared to these black ones, which are going to be susceptible mosquitoes, ones that will die if they're treated with an insecticide. What happens is they're very rare in our original population, but as we create selection events, as we force them down a particular lineage, in this case by treating with an insecticide, we get rid of most of the susceptible mosquitoes, but you can see compared to the now reduced population size, our resistant mosquitoes make up a larger proportion, which really gets amplified after the next generation of breeding. We see that there's now that ratio of resistant to susceptible mosquitoes has been maintained. And when we go out and treat the next time with the same insecticide, even more of those resistant mosquitoes survive, as opposed to most of the susceptibles now being gone. So they breed one more time, and after multiple treatments, this doesn't happen overnight, but it can happen relatively quickly. We have a population that is primarily composed of resistant individuals. I'd like to start today by talking about how to detect resistance. The main one that I'm going to focus on is the topical bioassay, which is what we do in our parish. There's also the genetic assay, the most famous of which is KDR, knockdown resistance. Acetylcholinesterase also happens as well, but we won't be going into that today. And finally, the bottle bioassay, which I know everyone is familiar with. You'll hear more about that in other talks, I'm sure, but not so much in this one. We're going to emphasize the topical because that is what I'm most familiar with. So if you're not familiar with the topical bioassay, it is a test where we apply a small dollop of insecticide to the thorax of the mosquito using this very small syringe. And it measures a very particular amount in grams of insecticide to kill your average mosquito, also known as the LD50. This is a little bit different than an LC50, a lethal concentration for the average of the population, because we know precisely how much insecticide this mosquito was exposed to, as opposed to a concentration. This is a brief video that we recorded a couple years ago, so I cringe at some of the things we now do differently, but this is how we do topicals. We'll knock them out briefly, we'll get them on a cold plate, extract some of the insecticide and solvent. This is me making sure that each click of our pipette is consistent. We'll grab a chilled mosquito and expose it to, I think it's something like 0.05 microliters of insecticide in solution. You can see this is a range of cups with different concentrations, or rather, and this is us weighing out the mosquitoes. It's very important to mass standardize everything, as larger mosquitoes are more capable of digesting smaller amounts of insecticide. You can see that they get cups in an incubator. I record some of our results. And this goes by quick, but there's a lot of dead mosquitoes on the left in our higher dosages, as opposed to the controls and lower concentrations on the right. So this is the data that we get at the end, and this is called a dose response line. It's complicated, I won't get into everything, but along the x-axis down here, we have dosage in nanograms of NALED in this case, or whatever your active ingredient is, per milligram of mosquito. This is logarithmic, so you can see it increases very quickly, and then percent dead on the x-axis, or the y-axis. And then this line here represents the LD50, the amount of insecticide it takes to kill your average mosquito from this population, the 50th percentile. In this case, it's 3.1. This does go how we would expect. The lower doses over on the left do have lower mortality than your higher doses over on the right. However, this by itself isn't very useful. It takes 3 grams or 3 nanograms of naled to kill an average mosquito. Where this becomes really important is when you have multiple populations. So this is that same line that you just saw in blue, but we've added another line for reference in green. This blue line is a susceptible lab strain of Culex squinx known as the Sebring population. This green one is one of our wild colonies that we've caught, I believe, in Slidell, Louisiana. And you can see that there's a pretty big valley between the two. It takes 3 nanograms to kill your average Sebring and about 50 to kill your average wild mosquito. This difference, the proportional difference between these two, is known as the resistance ratio. In this case, it's about a 20-fold difference between our susceptible population and our wild population. We want that number to be as small as it can be. 20 is not exactly great. So in St. Tammany, and in summary for the topical end of this talk, our average squinx require about 20 times more naled to kill. That number is increasing every year. It's now significantly more than that. This slide was written about a year ago. And as of about a year ago, 200 times more resmethrin to kill. That is a number that is approaching functional resistance as opposed to just a neat, scary finding that you have in the lab. So as a result of that, we have led to a couple of operational changes. We've introduced deltamethrin into our rotation. It still is a pyrethroid, unlike resmethrin, but at least it is a type 2 and has a slightly different mechanism of action, which we're going to talk more about mechanisms shortly. We've increased spray thresholds, so we're spraying a little less frequently, and we're also spraying less frequently because we require more time between our treatment intervals. So let's take a little bit of a shift and talk about resistance mechanisms. This is what underlies resistance in the first place, and they are random mutations that gave rise to resistant phenotypes or expressions of genetic changes. There's 3 different ones: target site mutations, amplified detoxification, and cuticle penetration. Let's begin with target site mutations. These are individual nucleotide mutations, or I think very occasionally locus of mutations, that result in changes in quaternary or tertiary protein structure. So if you remember back to biology class, you have something that's affecting change, your insecticide, and you have the receptor with which it binds to. Well, it binds normally very closely, but the moment you modify the receptor's shape just a little bit, and it can no longer accept that insecticide, the insecticide no longer functions. This is thought to be the primary means of pyrethroid resistance in 80 species, although there's plenty of evidence that there's these others as well. Amplified detoxification is exactly what it sounds like. It is also known as enzymatic resistance, and it is all about the mosquito producing enough enzymes to break down the insecticides it's receiving in order to sequester and excrete it such that it has no effect. And this is largely thought to be the primary mode of resistance in Culex mosquitoes. It certainly is in our mosquitoes, at least in St. Tammany. And finally, cuticle penetration. This one is kind of the most rare, or at least the least observed in nature as far as mosquitoes go. And it has to do with poor absorption due to the cuticle either hardening, getting larger, or just having a slightly different composition, which makes it so the insecticide doesn't penetrate as well. Like I said, this is not particularly common, and I doubt you'll hear a whole lot about this today. Otherwise, I would be very surprised. Let's talk about KDR, one of those specific base pair mutations. KDR, if you aren't familiar, is known as knockdown resistance, and that's because when these adulticides are used and you observe the mosquitoes and the effects, it causes temporary knockdown instead of death. So you might look after 5 minutes post-application and see that, hey, everything is dead. But if you come back a few hours later, you'll find that everyone is alive and well, unfortunately. It has a genetic origin at a specific set of base pairs on the sodium ion channel where pyrethroids and a few other insecticides function. And specifically, those mutations prevent the binding of pyrethroids. Like I say, they change the target site composition just enough that insecticides will no longer bind. The 2 most important, or at least the most studied, are a valine to isoleucine shift at codon 1016 as well as a phenylalanine to cysteine shift at 1534. There are many more of them, particularly if you look outside of 80 species, but those are the primary ones that have been studied in the literature. The one that I want to focus a little bit more on because it's closer to home, this is what we find a lot more frequently in St. Tammany Parish at least, is enzyme detoxification. We do detoxification in our bodies, primarily in the liver. I'm not a human anatomist, so I hope that I've got the right organ right there. But basically what happens, here's a very bad analogy for humans and then mosquitoes. We get a lot of bad stuff. Say you go out drinking one night, you've got a lot of alcohol in your body. Your liver recognizes, hey, this stuff is going to hurt you if it sticks around, so it breaks it down. In a weird world where evolution occurs very rapidly, what could theoretically happen, let's say the air composition changes, there's more toxic stuff in our air that we breathe, something might occur which causes selection for us to develop another liver. I know it's hidden by my picture, but something could happen over the eons that say, hey, in this animal that's exposed to a whole lot of bad stuff, maybe they need a second liver. That's probably not going to happen in humans, but it very well is something that has, I'm sure, occurred throughout history in other organisms. If we change that slightly and make this maybe a little bit more realistic, in mosquitoes and at insects in large where you have a whole lot of generations rapidly, what can occur is selection for amplified enzyme activity. So maybe they're not growing a second liver because they don't have one, but it very well could occur that their enzyme activity gets ramped up. At the downside of something else, maybe they're worse flyers, maybe they have less energy for something else. But in this instance, if we kind of take it through the liver example from earlier, if there's a bunch of organophosphates that these mosquitoes are being exposed to, selection could occur such that only the mosquitoes that are able to sufficiently break down that organophosphate are the ones that are surviving and reproducing. The way that we test for this in our lab is through a spectrophotometric assay. We call it an esterase assay. It's specifically looking at alpha esterases. We're using alpha naphthyl acetate as a stand-in for an ester insecticide, which a lot of pyrethroids and OPs consist of. And this is what it looks like. So the deeper the color, so all of these wells in this 96 well plate contain some mosquito homogenate. We ground up mosquitoes and put their guts in here, as well as we're introducing a standard substrate that it could break down and see how well the mosquito does at breaking that down. The deeper the color, you can see these really dark purple colors here, the more esterase activity that we find. So the more insecticide breakdown occurs in these deeper, darker wells. So the result of this might be something along the lines of this. This is what we found from a study back in 2021 in St. Tammany, looking at esterase activity in different regions around our parish. So along the x-axis, we have different populations from across our parish. Sebring is that population that I mentioned earlier that's a lab susceptible colony. They're very wimpy. We would expect that they have low esterase activity. The y-axis, don't worry about the unit because it's horrible, but this is the amount of enzyme detoxification activity, specifically esterases in this case. You can see that there's a few populations, Slimmer and Covington, for example, that have a very wide variance. There's certainly mosquitoes that have high activity, but there's also ones that have low. But then you look at places like Lacombe and Ravine, which I believe is also in Covington, and you see that they are significantly different. There's very little overlap between the Sebring population and the 95% confidence intervals for either of these populations. So we're seeing amplified detoxification in at least a few of our populations in St. Tammany. To wrap up this part of the talk, we're going to kind of change directions after this. Resistance is already out there. You might think, no, we don't have resistant mosquitoes in my district. You do, you just might not have very many of them, although I would argue you probably have more than you think. The real question isn't, is resistance there or not? It's how well are you selecting for it and curbing its development over time. If you have a large Aedes aegypti population or Aedes albopictus, and that's what you do a lot of your spraying for, KDR is a likely suspect as far as what you might need to be looking into as far as mechanisms go. If you find plenty of resistance with KDR, your solution might be to rotate with an organophosphate. We don't tend to see as much KDR effect on OP usage. If you have a lot of Culex mosquitoes like we do, and you have Culex resistance, there's a good likelihood that enzymatic detox is what's to blame. And the solution, similar to your Aedes species, is to rotate. Use a different mechanism, or if you have to keep using that same pyrethroid, at least introduce a synergist to minimize the ability for enzymes to break down that insecticide. And finally, mosquitoes don't know any borders. Even if you're doing everything right, that doesn't mean that agriculture down the road is performing everything up to snuff, or that your neighbors might not be having the same problems, which I think is a good lead-in to the LMCA, Louisiana Mosquito Control Association, statewide resistance program. I'd like to just spend a few minutes on this as the committee head for this program in Louisiana. Our intent is to build a statewide resistance database. This is useful for historical reasons, to be able to see how things change over time. It also informs if your neighbor's mosquitoes might be hurting your own control efforts, like I mentioned before. And secondly, and probably more importantly, to provide resistance data for smaller parishes that maybe don't have the funding or the skill set to do these kinds of tests. Currently, it is funded by the state. Our Department of Health has granted us grant money for the last couple of years, and we hope going forward. It is performed at a central university to kind of make it as convenient as possible for remote districts to participate. This is currently at LSU, Louisiana State University, in East Baton Rouge. And they historically had done KDR genetic testing. They currently do bottle bioassays, and all of this testing was only performed on Culex squinks. We might expand that to Aedes in the future, but we've got a whole lot of Culex issues in Louisiana. So to touch on our KDR participation, we had over 9,000 mosquitoes tested in 2020, the first year of KDR testing, and it was a huge success until in 2021, we had 0 mosquitoes tested. And that was intentional and unfortunate because the USDA out of Gainesville in Florida, who we were closely conspiring with to get all of this set up, published some research saying, hey, Culex resistance and KDR are poorly linked. When we see high rates of KDR mutations, we don't necessarily see high phenotype resistance. So as a result of that, we culled it. It's not useful to have a test that's somewhat expensive and laborious if there's a very poor link. As I mentioned earlier, Culex resistance appears to favor enzymatic mechanisms. That's what we find in our parish, and that is backed up by what the USDA found. There are exceptions, of course. We had about 30 bottle bioassays performed in 2020, which was not great for our 60 some odd parishes in Louisiana, and that dropped to less than 10 in 2021. Not a huge success. It's apparent why. It requires a lot more labor. You don't just ship in pools of dead mosquitoes. You actually have to go out and collect living mosquitoes, rear them, deliver them potentially a few hours away to the central location, which has led to a couple of major changes in how we're doing it in the coming years, considering this lack of participation. What we're doing this year into next year is contracting with a local university to hire out some of their students for insect collection, rearing, and delivery. The plan is for them to visit a couple of parishes per week in May through July and retrieve either larvae if they can find them, or at least egg grafts if nothing else, and then bring them back for the bottle bioassay. In the future, I would love to use topical bioassays. I think it's a much more sensitive and discrete test than the bottle bioassay, but I'm more than happy to use any test that we can use to get some resistance data from across the state. And secondly, I'd love to be able to test for enzymes. So this will give the people who are getting this data a little bit more direction, as opposed to just saying, hey, you've got resistance problems, good luck. This says you've got resistance problems, but it looks like this synergist might be a good fit for introduction if you want to get back to normal. So some takeaways: don't start an in-depth new collaboration during a pandemic. I don't recommend it. There were definitely some problems there. Participation is not guaranteed. Shipping worked great for KDR, but the moment we lost KDR, it didn't really work well for bottles. And if you want IR data, you're probably going to have to go out and collect it yourself. It's going to cost money. If you're expecting free labor, that is a rarity, is what we discovered. With that, I'll take any questions at my email and delisi@stitmad.org, and I look forward to speaking with everyone at the roundtable. Thank you for your time.